Hello everyone, today I am honored to introduce blood biomarkers of Helicobacter pylori-infected chronic gastritis in active versus stable phases, a clinical indicator analysis combined with proteomic and metabolomic investigations. Addressing the limitations of gastroscopy-dependent diagnosis for H. pylori gastritis, this study integrated multi-omics data to reveal three core molecular mechanisms of active disease. We successfully constructed a non-invasive combined diagnostic model comprising hemoglobin, CAPZB protein, and acetic acid, which achieved an AUC of 0.9978 for distinguishing disease stages. This provides a revolutionary tool for the precise and dynamic management of chronic gastritis. Highlights. Based on blood test data of 1409 real patients, K-means unsupervised clustering accurately stratifies gastritis into active and stable phases, delivering a convenient non-invasive evaluation method, combines serum proteomics and metabolomics decode molecular pathways, clarify the inflammatory vicious cycle of hematological dysfunction, energy reprogramming and cytoskeletal migration, and screen stage-specific biomarkers simultaneously. A cross-omics panel, HGB-CAPZB acetic acid, achieves an AUC of 0.9978 for staging. Molecular evidence proves stable phase low-grade chronic inflammation, laying theoretical foundation for gastritis relapse prevention. Analysis of clinical blood biochemical indicators. A total of 1409 patients with H. pylori-infected chronic gastritis were enrolled. 6 core blood biochemical indicators, HGB, UA, CK, RDWCV, CHE, GGT, with extremely significant intergroups, p less than 0.001, were screened via unsupervised clustering. In active phase patients, HGB and CHE were markedly decreased, while RDWCV, UA, CK and GGT were elevated, which can effectively differentiate active and stable gastritis. Serum proteomic analysis. Serum proteomics was performed on patients with active/stable gastritis and healthy controls to screen stage-specific differential proteins. PPI network analysis verified core molecules including CAPZB, TFRC, CFL1, ACTR2/3 and ENO3 regulate cytoskeletal remodeling and immune cell migration during gastric mucosal inflammation, offering protein biomarkers for disease staging. Serum metabolomics analysis. Untargeted metabolomics was conducted on serum samples from 3 groups, and 13 core metabolites with strong discriminatory power were screened. Acetic acid acts as a key marker. Disorders of carbohydrate, amino acid, and energy metabolic pathways exist of gastritis, and energy metabolism reprogramming serves as a vital molecular feature to distinguish disease stages. High-precision diagnostic model for active and stable stages of gastritis. A triple biomarker diagnostic panel, HGB-CAPZB acetic acid, was established integrating clinical, proteomic and metabolomic data. It reaches an AUC of 0.9978 to differentiate active and stable gastritis, enabling non-invasive and dynamic disease staging. Summary. Accurate classification of clinical indicators, using unsupervised K-means clustering algorithm to analyze routine clinical blood test indicators. Patients with Helicobacter pylori gastritis were successfully divided into active and stable phases. Deep decryption of multi-omics mechanisms. Linkage analysis of proteomics and metabolomics reveals the core molecular pathological network of active stage lesions. High-precision cross-omics model. A cross-omics diagnostic panel of HGB plus CAPZB plus acetic acid was extracted from the 3 core biological pathways, achieving an AUC efficiency of 0.978 in distinguishing between active and stable phases.